Efavirenz falls under a class of anti-HIV drugs known as non-nucleoside reverse transcriptase inhibitors, abbreviated as NNRTIs. To understand how efavirenz works, let's first re-explore what happens in our cells once HIV has infected and entered our cells. Remember that HIV is an RNA retrovirus. The RNA means that instead of being double-stranded like our normal DNA, the HIV genetic code consists only of one strand. The retrovirus means that the HIV RNA requires an enzyme to complete the other half of the viral RNA to form the proviral DNA. The proviral DNA is then what hijacks our host cell DNA and creates a blueprint for the rapid proliferation of HIV. The enzyme required to convert HIV RNA to proviral DNA is called reverse transcriptase. Reverse transcriptase is included in the viral RNA package that was delivered into the cell. Let's now look at how reverse transcriptase works to create the proviral DNA. Reverse transcriptase has a pocket where it binds to the viral RNA, called the active site. Reverse transcriptase binds to the viral RNA and goes along the single-stranded RNA and adds the complementary half to form the double-stranded proviral DNA. Efavirenz treats HIV by targeting the reverse transcriptase enzyme. Efavirenz is a non-competitive inhibitor of reverse transcriptase. This means that efavirenz targets and binds to a distinct site on the reverse transcriptase that is different than the site that binds to the viral RNA. This site is called the NNRTI pocket. Efavirenz binds to the reverse transcriptase, and this causes the reverse transcriptase to change shape, and more importantly, changes the shape of the active site that binds to the viral RNA. The change in shape of the reverse transcriptase after the efavirenz binds to the enzyme is significant enough that the reverse transcriptase can no longer bind to the viral RNA. Thus, the viral RNA is blocked from being converted to the proviral DNA that would then hijack our host cell's DNA. Let's now look back at our host cell. The binding of efavirenz on the reverse transcriptase blocks the viral RNA from being converted to the proviral DNA. Thus, HIV cannot hijack our host cell DNA and insert the instructions for HIV into our blueprint. This helps control the amount of HIV in our blood, otherwise known as our viral load. Our viral load is controlled because we've reduced the number of host cells that are rapidly creating the HIV that will then in turn infect more of our host cells, causing the rapid proliferation of HIV,